Good afternoon, and thank you everyone for coming and for taking this time in the early afternoon to come to the Gait and Brain seminar. Today we are very fortunate. We have Dr. Louis Lipsett for the Harvard Division on Aging. I would like to do later the formal introduction of him. But at the beginning I have one or two slides just to do a very brief overview about the Gait and Brain seminar. We started with this idea 3 years ago just to highlight the importance in geriatric research about the relation between gait and mobility problems and cognition, particularly because in the last 20 years all the research focused on mobility sometimes didn't address the cognitive issues, and vice versa. When we did a lot of research in cognition and dementia, we didn't address the problems about mobility, falls. And in our practice, we know that both problems are, in general, very prevalent and they co-occurrence. Particularly, we know that mobility decline and cognitive decline are two key features in the aging process, and in general, they overlap, right? Particularly, we know the fact that falls and mobility issues are twice more prevalent in people with cognitive decline and dementia than cognitive normal people. So that was the idea where we started with this kind of seminar just to highlight the research. I was very fortunate when I started my training in the first part of my PhD in gerontology, when I did the elective with Dr. Louis Lipsett at Harvard. He was very nice to accept me in his lab at Harvard. And I started to learn with him all the complexity of the old people. And we came up with the idea about that we call at the moment the elephant syndrome. Basically, it describes that a disease afflicting many scientists who miss the significance of their findings because they do not grasp the entire situation. So it's like the 5, 6 scientists, blind scientists, and the elephant. So one scientist may grasp one part of the elephant, thinking that that part of the elephant is a wall, or they may grasp a leg of the elephant and may think it's a column, or they may grasp other part and think it's a tree. So sometimes that happens in geriatric medicine. Sometimes we focus just on one aspect of the elderly patient and we miss the whole picture, and or perhaps we cannot grasp the entire situation. Particularly, Dr. Louis Lipsett, with his study of complexity of the aging process and frailty, I think he was one of the first that put in numbers, in research, the complexity of the older people and ways in where we may grasp the complete situation of people. And in the recent years, he came up with hypotheses and ideas about syndromes in the aging population, when we see some prevalent diseases that we think that are normal or associated to aging. Sometimes you have to be a little challenging going against the current to say, no, that is not normal, it's very prevalent, and we can find something behind those problems. So that's the reason we invite Dr. Lipsett to give the lecture about a syndrome that he described as the frontal subcortical syndrome of the aging process. Some housekeeping things before starting with the presentation of Dr. Lipsett. I really want to thank Jennifer. Jennifer helped me a lot in the organization of this seminar, so I really thank to her. And also we got funding from the Campbell Lectureship Fund for the Department of Medicine just to bring Dr. Lipsett here, also for the Gait system. So thank you very much for the support. Particularly, just a quick introduction about the bio of Dr. Lipsett. He's professor of medicine at Harvard Medical School and chief in this moment of the gerontological division at Bethany Radicons Medical Center, and co-director of the Institute of Aging Research. Also, he has served before as principal investigator of the Harvard Foundation Center for Excellence, and also he has run in several projects funding for the National Institute of Health and the National Institute of Aging. Particularly in the last few years, he got funding for a very important study that he called Mobilize Boston, which is a cohort study, community-based, which includes almost 100 participants from the community, studying features about demographics, social mobility, vascular risk factors. Part of his presentations, he's going to present that data. Other thing that I really want to highlight from Dr. Lipsett is that he's one of the first geriatricians that put geriatric medicine in the big journals. In the 80s and the 90s, he was one of the first geriatricians published in New England Journal of Medicine, JAMA, Annals of Internal Medicine, describing really, really important geriatric syndrome. For example, he was the first to describe postprandial hypotension. Before, people didn't care about older adults, you know, fainting after having the breakfast in nursing homes, and Dr. Lee said, no, something is happening here, and he came up with a new syndrome, and he published in New England. So I think we owe you as a geriatricians to put geriatric medicine in the big journals in the 80s. Thank you, Lou, and thank you for coming today.
Well, thank you, Manuel. I didn't know who you were talking about, though, but I appreciate your confidence. Well, it's been a real pleasure visiting London. I guess it's the real London. And meeting all of you and a variety of people in this very impressive organization and faculty. What I'd like to do is, in as informal a way as possible, share with you some observations that we've made that I think will resonate with many of the geriatricians in the group, and hopefully leave plenty of time for discussion at the end. So I'm going to talk about the what we've dubbed the microvascular frontal subcortical syndrome of aging. And before I get into this too deeply, I've been told I have to beat this up, I guess, to get it to go. Whoops.
I'll just reset it.
Oh, you'll reset it? Okay, maybe we'll do it that way. Well, it's not happy. There we go. So I was told that I need to have a disclosure statement, which we do, and basically I have no conflicts of interest, so enough of that. Well, I'd like to begin with this picture of an older person and ask the question, what makes a person look old? There are many characteristics, but one of them is slow gait. Another, perhaps, is mental slowing. And a third is depressed affect. And I think this gentleman on the left probably demonstrates all three of those. Well, what do these symptoms have in common? One of the things I'd like to elaborate on today is that I believe they all have these white matter hyperintensities in periventricular and subcortical regions of the brain in common. This is a very common finding that you'll see on CT scans and MRIs on a vast majority of our elderly patients. In the past, radiologists would write this off as an incidental finding, quote, consistent with age, of no particular significance. And I'd like to argue that, in fact, I think it has tremendous significance. In fact, it is probably these changes that underlie many of the symptoms that we come to consider characteristics of aging. Well, these white matter abnormalities that I've shown you on this MRI are in regions located right around these ventricles where there is relatively little perfusion. These are called watershed areas of the brain. And you can see here in this nice MR angiogram the large vessels, the carotids coming up and branching into the middle cerebrals, and branches upon branches go into these little spaces here until there's virtually nothing surrounding the ventricles. And these are areas that are highly vulnerable to hypoperfusion. And as a result of hypoperfusion, damage can occur that results in these white matter changes. Here's another example of that, where you see the carotid arteries branching into the middle cerebral, and then these perforating branches into these periventricular white matter regions. And these are exactly the regions where these white matter changes occur on the MRIs of many of our older patients. If one looks at this under the microscope, you see here the MRI picture on the left with these white matter changes. Whoops. And then under the microscope, in a normal brain, you'll see these tiny little vessels which drop out in those regions and are replaced by gliosis and inflammatory changes in the patients with white matter change. If you do an autopsy and look at the brain under the microscope, similarly you see this pallor and loss of neuronal tissue, which corresponds to the loss of blood vessels in those regions. So we do think that much of this relates to ischemic damage or inadequate oxygen delivery to those parts of the brain. So where do white matter hyperintensities come from, and what harm do they do? This slide illustrates the sequence of events which I believe might underlie the development of these changes and subsequent symptoms. So it all begins with cardiovascular risk factors that occur predominantly in middle age. And these, of course, include hypertension, diabetes, hyperlipidemia, and other risks. These eventually affect blood pressure regulatory mechanisms that result in cerebral hypoperfusion. This can result in ischemic damage to these watershed regions called cerebral microangiopathy, which can ultimately cause the frontal subcortical symptoms that I've begun to describe to you, such as executive dysfunction, a form of cognitive dysfunction we'll discuss, slow gait, and depression. Now, what is the evidence that any of this exists? Well, there is, in fact, evidence, shown here in the red 1, that cardiovascular risk factors lead to cerebral hyperperfusion. There's evidence, too, that cerebral hyperperfusion leads to cerebral microangiopathy. We'll review some of this. There's evidence that cerebral microangiopathy is associated with frontal subcortical symptoms. And there's evidence that cerebral hyperperfusion is linked to these symptoms. And finally, that cardiovascular risks also are linked to these symptoms. Now, much of this work is cross-sectional and allows us to piece together a puzzle, but we really require some longitudinal studies to prove that this, in fact, takes place. And I'll give you some examples of what we know and what we don't know in this cascade. Well, first, let's start with cardiovascular risk factors. Hypertension is perhaps the most prevalent, and this slide illustrates the increase in the prevalence of hypertension with advancing age. Shown here is the percent of the population with hypertension from 20 to 34, all the way up to 75 plus. And you can see that in men, in the blue, and in women, in the red, there's a dramatic linear increase in hypertension, such that by age 75, nearly 2/3 of men and 3/4 of women have hypertension. So this relationship between age and hypertension makes it very difficult to distinguish what is due to aging and what is due to blood pressure elevation. So let's look at that. Here we have a schema that shows how both aging and hypertension can influence the regulation of blood flow to the brain. Aging, whoops, let's go back. Aging, shown here, actually impairs blood pressure regulation. Aging, as I've shown you, is associated with blood pressure elevation, and blood pressure elevation further impairs the regulation of blood pressure. With abnormal blood pressure regulation, we can develop periods of hypotension during activities. This can lead to cerebral hypoperfusion, particularly in watershed regions, which can then lead to ischemic damage and some of the symptoms that result. So what is the effect of aging on blood pressure regulation? Well, there are many different changes that occur in the physiology of blood pressure regulation. But the first and perhaps the most important is an impairment in the baroreflex. As many of you know, the baroreflex is a very important mechanism that enables our body to maintain a relatively stable blood pressure. If pressure falls, we increase our heart rate and our vascular resistance to restore it back to normal. If blood pressure goes up too high, we slow the heart and dilate the blood vessels to bring it back down. Well, this ability to regulate blood pressure through the baroreflex is impaired with advancing age, so that older people have much more blood pressure variability and greater susceptibility to hypotension. Also, the second point here is that the older kidney loses some of its ability to hold on to salt and water. And as a result, it tends to waste sodium and fluids, and older people readily become dehydrated. And this can result, again, in hypotension. Thirdly, the older heart gets stiff with age. It doesn't relax very well. Therefore, it doesn't fill with blood very well, and cardiac output can fall. So again, this is another reason why blood pressure may drop when volume is reduced or when people are not eating or drinking as much. And finally, there is a baseline decrease in cerebral blood flow in older people, so that you add all of these things together of reducing perfusion to the brain. So this is what happens with aging. At least we think today, normal aging does this. But now if we add on top of that stiffening blood vessels. Hypertension further impairs diastolic function of the heart and the ability to relax and fill with blood. Hypertension increases the threshold of regulation, making older people more vulnerable to hypoperfusion at even higher blood pressure levels. So you can see how the two, aging and hypertension, conspire to make it very difficult for us to maintain a relatively stable blood pressure. Well, we asked whether all of these physiologic considerations really were significant in a clinical setting many years ago, and did a study illustrated here in which we actually measured systolic blood pressure, shown on the y-axis, during a standardized series of activities, shown up here, between the hours of 7 a.m. and 1 p.m. in two groups of people. Older people living in our long-term care facility. These are people like you see here in the rehab component of this building, and healthy young people. These were employees and medical students and residents and fellows. We had them, everybody sleep overnight, and then we had them first measure their pressure, first supine in the morning, then after standing and doing their personal hygiene, they sat for breakfast and ate a large breakfast and then remained seated for an hour. They then stood and ambulated. Later they took a nitroglycerin tablet, that should be NTG. Then they stood, they ambulated, they took their usual medicines, ate lunch, and stood again. And I think you can see a couple of things here. First, the older individuals started with much higher blood pressures. They were relatively hypertensive, as we would expect. And then the older folks had marked variability in response to these activities. The young had some variability, but certainly not as much. And look what happened after some of these activities. They dropped their pressure when standing. Look at this, blood pressure plummeted after breakfast, after standing and ambulating, after nitroglycerin, it fell markedly, and then after medications and lunch. Now these things did not happen, those hypotensive episodes did not happen in the young people. So I like to ask, what do standing up, eating a meal, and taking nitroglycerin have in common? Sorry? Yeah, to some extent. Any other? What else do they do? Well, they dilate parts of the body that pool blood, and as a result, there's less venous return. There's preload reduction. Now, in response to this preload reduction, what should the body do? What should the baroreflex do? Sorry? Residents know the answer to this, right? Well, it should. Vasoconstriction, and what does the heart do? Increases its heart rate. So we'd expect an increase in heart rate and vasoconstriction to compensate for this preload reduction. So let's look at what happens to heart rate. Here's the same slide, only now we're looking at heart rate on the y-axis, and the curves are opposite. Here, the young have the marked variation in heart rate, and the old have the blunted response. That is the blunted baroreflex I've been talking about. So the young increase their heart rate when standing, after breakfast, when standing and ambulating, after nitroglycerin, and after lunch and medications. So the young can compensate, but the old can't. And as a result, the older individuals have these periods of hypotension throughout the day. Well, one other take-home message I'd like to leave with you is the notion that the higher you are, the farther you fall. So it is the hypertensive older people who have the greatest risk of hypotension. It seems sort of paradoxical, but it's illustrated here where we've measured, we've measured the systolic blood pressure change after a meal, as a function of the baseline blood pressure that a person had. And you can see those people with higher blood pressures have the greater decline in blood pressure after eating. And the same is true of orthostatic blood pressure change. The higher they are, the greater the orthostatic blood pressure change when they stand up. So this is very important to recognize that our hypertensive older people are at greatest risk of hypotension. Well, the body is too smart for that because it wants to maintain a relatively stable cerebral blood flow even in the face of hypotension. And that mechanism to maintain a relatively constant cerebral blood flow is called cerebral autoregulation. And that's shown here. Here we're plotting the cerebral perfusion pressure, or think of this as just the blood pressure, and cerebral blood flow. And in the normal autoregulation curve, there is a plateau, so that even though there are changes in blood pressure from about 80 systolic up to about 180 systolic, cerebral blood flow remains constant. Below a certain value, though, blood flow begins to drop, and above a certain blood pressure, blood flow begins to increase, which are, of course, danger zones for the brain. Imagine that you are supine and your blood pressure is around, 140 or so, and then you stand up and your blood pressure drops to 120 or so. You're still within this range. Your brain doesn't see any change in blood flow. Well, now, what happens with hypertension? Hypertension shifts the curve to the right. So now we're regulating blood pressure at higher values. So that same supine to standing maneuver now in a hypertensive individual might tip the balance so that when standing, cerebral blood flow falls off. This is what I meant earlier by increasing the threshold for cerebral autoregulation. Now, the good news is that if you treat hypertension, you begin to restore this curve back toward normal. So treating hypertension can actually improve cerebral autoregulation. Another good reason why we want to treat hypertension. All right, well now let's go back to that diagram that illustrated a sequence of events from risk factors to symptoms and look at what evidence there is supporting each part of the pathway. So the first part of the pathway is the association between cardiovascular risk factors and cerebral hypoperfusion. What is the evidence? And unfortunately, there's mostly cross-sectional evidence between, showing this relationship between risk factors and cerebral hypoperfusion. Here is an example of one study that was done by my colleague Vera Novak at Beth Israel Deaconess Medical Center, in which she used arterial spin labeling, or CASL, with a 3 Tesla MRI to measure the brain's response to breathing carbon dioxide. When we breathe carbon dioxide or it increases in our bloodstream, our cerebral vessels dilate and we increase blood flow to the brain. When we hyperventilate, those vessels constrict and we reduce blood flow to the brain. So this is a nice way to test how well the brain regulates perfusion in response to a chemical stimulus like carbon dioxide. So here, across the top, are 3 control subjects. The yellow shows blood flow. The intensity of yellow shows intensity of blood flow. So here under baseline conditions, all of these subjects had some blood flow here in yellow. But when they breathed carbon dioxide, blood flow increased. And when they then hyperventilated to reduce carbon dioxide in the blood, blood flow decreased. And then later, when they equilibrated, they came back to baseline. On the bottom are 3 hypertensive subjects. And you can see that they don't have much change in cerebral blood flow in response to hyper or hypocapnia. So this illustrates the impairment in the regulation of blood flow to the brain that we see in hypertensive patients. Well, the same is true of diabetes. Here the 3 subjects are vertically arranged. So here we have 3 control subjects vertically and 3 diabetic subjects. And again, they go from a baseline to breathing carbon dioxide, and you can see the increase in blood flow. Then they hyperventilate, and then there's a nice decrease in blood flow, and then they begin to re-equilibrate. In the diabetics, despite the breathing of carbon dioxide or hyperventilation, there is no change in cerebral blood flow. So diabetes also impairs this important mechanism of blood flow regulation. Okay? So what is then the evidence for the association between cerebral hyperperfusion and the development of white matter lesions in the brain? And here we have some cross-sectional data and longitudinal data. Listed here are some of the cross-sectional studies. For example, the Rotterdam SCAN study. This study looked at 892 subjects aged 60 to 91, and they found that low brain perfusion was associated with higher white matter lesion volume. In the SMART study, published in 2004, 228 subjects with cerebrovascular disease also showed that total cerebral blood flow, the higher the blood flow, the lower the prevalence and the lower the severity of white matter lesions. The Copenhagen study, similarly, among 85-year-olds, showed that lower cerebral blood flow and lower reactivity to carbon dioxide resulted in greater amount of white matter hyperintensities. And if you actually go and under the microscope and look at the brains of people with these low blood flow and white matter changes, you actually see staining for proteins that are related to hypoxia or related to low oxygen and ischemia. One of these proteins is called HIF-1, or hypoxia-inducible factor, which I'll talk about in a few minutes. So I think there is evidence for an association between hypoperfusion and white matter lesions. But some of the best evidence comes from the recent PROSPER study, which is longitudinal. In this study, they examined subjects, this was done in Denmark, for the development of white matter hyperintensities and correlated this with the change in cerebral blood flow over 33 months. So cerebral blood flow was measured using gradient echo phase contrast MRI. And they did it at baseline, and then they measured white matter again 33 months later and correlated it with the change in cerebral blood flow. And here you see that there are the odds ratios for the development of white matter hyperintensities and divided by different regions of the brain. And you can see that in the periventricular regions, a change of cerebral blood flow, or a decrease by 50 ml per minute, resulted in a 23% increase in the development of white matter hyperintensities. Here, when adjusted, there was actually a 32% increase in white matter hyperintensities over 33 months related to a 50 ml per minute decrease in blood flow. So this was pretty convincing evidence that a reduction in blood flow predicts the further development of white matter change. Okay. We've done some other cross-sectional studies here in diabetes where we've correlated continuous grade of white matter or the white matter hyperintensity volume with blood flow velocity using a transcranial Doppler procedure. And once again, the lower the blood flow, the higher the grade or volume of white matter hyperintensities.
Could that not be the other way around?
In other words?
Hyperintensities, you need blood flow?
Well, in other words, is it because of the loss of tissue that you reduce blood flow? Absolutely, and that's why the longitudinal study was so important, the PROSPER study that showed that prospectively the reduction in blood flow was associated with a greater increase in the white matter. Now, we don't, again, you only have 2 points in time, so you really don't know exactly when that took place. I think that question still remains. Is it due to a loss of tissue, or is low blood flow actually causing a loss of tissue? But I think the PROSPER study is the first to begin to suggest that it may be causing a loss of tissue. But I agree that we don't know that for sure. So thirdly, what is the evidence for the association between cerebral white matter lesions and the development of symptoms? And the three symptoms in particular I mentioned at the beginning that characterize many older people are alterations in cognition, particularly executive dysfunction, which is the ability to plan, organize, multitask, and sequence activities. Changes in gait, in the case I talked about earlier, slow gait in particular, and changes in mood, mostly depressive symptoms. And there now is evidence from the lattice study that severe white matter lesions are associated with cognitive and functional decline over 1 year. There's evidence from Whitman and others that in subjects over age 74, that declines in Tinetti scores over 4 years are associated with an increase in volume of white matter lesions in the brain. And in the Rotterdam study, among almost 2,100 subjects over average age 72, depressive symptoms were associated with impairments in blood flow measured by transcranial Doppler or with impairment in CO2 reactivity. So it seems that there might be, there's associations between cerebral white matter lesions and alterations and symptoms. Here's just one example of the relationship between these white matter lesions and gait abnormalities. The topmost MRI series illustrates a normal individual over, I think, age 80. And here you can see in this three-dimensional diagram some of this white matter change illustrated in yellow. And here's someone with a gait disorder, a slow gait speed. And you can see here a greater amount of white matter hyperintensities, which are exaggerated here in yellow and look quite prominent here in this three-dimensional rendition. So we do think that, again, slowing of gait is associated with the white matter hyperintensities. More recently, actually this year, Srikanth from Australia published a very important paper in Stroke that indicated that these white matter lesions may be prospectively related to falls. What he did in his study is he divided the white matter volume into quintiles from the lowest volume of white matter lesions all the way up to the highest, and looked at the percent in each quintile who developed falls over a period of, I think, 1.5 years or so. And you can see that those in the highest quintile of white matter lesions developed, had the greatest percent of falls, or the greatest number of fallers. Here are incident falls as well, multiple incident falls, and again, the highest quintile was associated with the greatest percent of individuals with falls. So in gait scores as well, you can see a relationship between the quintiles of white matter lesion volume and the gait scores. Those in the highest quintiles had the lowest gait scores, and similarly in gait variability, those in the highest quintiles had the highest gait variability. So this again suggests that relationship between white matter lesions and some of the symptoms of aging, namely falls and gait disability.
Do you feel this is reflecting some sort of threshold, or does it actually matter where the distribution of the lesions are? There's certain areas which are more vulnerable in the same way that we think of strategic infarcts and its effect on cognition. Are there certain areas where you're more likely to...
Yeah, not in this study, but in some of the studies, particularly those looking at cognition, it's the periventricular regions that are probably more associated with executive dysfunction and some slowing of gait. And these are regions taking fibers from the frontal cortex. So I think region is important, though that has not been well worked out, because in some of these analyses where you have more of a qualitative measure of what the grade of white matter is, it does not distinguish the location. And now that we can actually define regions of interest, studies are beginning to emerge that are looking at the regional changes. The deep subcortical white matter may be well infarct related, more the lacunar infarcts, while the periventricular is probably more this ischemic damage in watershed regions. So, but that's a very important question.
Also, there is a study published last year, I think it was in Arca, if I'm not making, where correlate white matter disease with hip fractures.
Is that right?
Yes,
yeah.
Hip fracture.
Well, it certainly makes sense. Okay, so what is the evidence of association between cerebral hypoperfusion and frontal subcortical symptoms? Again, there is some evidence here, but most of it comes from work in the area of neurovascular coupling. I think it's important to understand this principle, which basically says that cerebral blood flow increases to meet the energy requirements of neuronal activity. So when we walk, when we talk, when we think, when we do some cerebral activity, we need to increase blood flow to that region in order to provide oxygen and metabolic substrate for the neurons that are functioning in that region. Well, we can measure neurovascular coupling using transcranial Doppler ultrasonography that can detect increases in cerebral blood flow to specific regions. And we like to look at it in the middle cerebral artery and frontal cortex, which are regions related to sort of frontal subcortical activities. And some of these frontal activities include gait, but also executive function. So we can actually look at the blood flow to the frontal and parietal cortex during tasks that involve executive function. And one of those tasks, a frontal executive task, is called NBAC, which requires the volunteer to identify a letter that occurred 1 or 2 back from the one that they're currently looking at in a sequence of letters. So here's an example. If you give this series of letters to an individual, they need to identify the B because that appeared 2 prior, or the L because that appeared 2 back, or D, that appeared 2 back. That would be called a 2-back test and requires executive functioning. So we measured transcranial Doppler blood flow through the middle cerebral artery in this case, using this probe and headband while people performed the 2-back test. And during this very nice test, we can actually demonstrate that there is an increase in cerebral blood flow at the point of the arrows here when they're doing the test, and that blood flow decreases when they're resting, and it can be reproduced over and over again as they perform this test. So we were interested in the percent increase in cerebral blood flow as a measure of neurovascular coupling. We actually wanted to see whether neurovascular coupling was impaired in people who had slow gait speeds. So we recruited 2 groups of people, one with fast gaits, 14 people, and one group with slow gaits, 22 people. This is still work in progress. People with slow gaits were a little bit older, but otherwise their characteristics were not statistically different, although there were tendency for those with slow gaits to have more cardiovascular risk factors. But their gait speeds were dramatically different. These people walked at about 1.1 meters per second on a 20-meter track, and these at 0.57 meters per second. So there were clear differences in gait speed. And then we looked at the percent activation of cerebral blood flow during the NBAC testing. The 1-back test was not a very strong stimulus, so we did not really get significant differences, although there were trends here. But when we did the 2-back test, we did find significant or nearly significant differences. And here is the percent change in blood flow velocity for the 2 groups. And you can see the slow people increased their blood flow velocity by about 2%, while the fast walkers by about 6%. And this was just on the verge of significance. And the number of correct responses here were also measured. On the 2-back test, the slow walkers got about 74% correct, while the fast walkers got 89% correct, which is highly significant, and suggests that not only are they performing better in an executive function task, but they're also walking better and activating the brain better during this particular task. So we do, again, think that this is evidence that alterations in the ability to regulate blood flow to the brain may be related to some of the slow gait speed and executive dysfunction that we see in our older patients. Okay? We are also asking whether cerebral hyperperfusion is associated with slow gait and falls in our Mobilized Boston study, which Manuel mentioned earlier. This is a 5-year NIA-funded project that we are just finished 5 years of in 800 people living in the Boston area within a 5-mile radius of our hospital. The age is actually over 70. The average age is about 78. In this study, we gathered very careful baseline assessments and repeated those assessments 18 months later. Half of the 3 hours of the assessment was done in people's home and 3 hours in our laboratory. And we measured everything that we could think of: medications, pain, memory, strength, balance, foot problems, but most importantly here, blood flow to the brain during CO2 inhalation and in some other circumstances. So we looked at the brain's response to carbon dioxide using a Doppler procedure. And then we followed these people monthly for falls by asking them to fill out a falls calendar, where on every day they would indicate they fell or didn't fall and send that calendar to our office. If they didn't send it in, we'd call them and track them down until we got the answers. So we know pretty well that these are accurate records of fall rates. And here are some of the data. First, we looked at the relationship between this cerebral vasoreactivity to carbon dioxide and falls. We divided our vasoreactivity into 5 quintiles, shown here, from the lowest to the highest CO2 reactivity, and then looked at the number of falls within each quintile over a follow-up period of about 2 years. And here you can see that those people in the lowest quintile of vasoreactivity had the highest fall rates. This was not quite a linear relationship. We don't know quite why this third quintile was so different, but in fact there was a general trend such that those in the highest quintile of vasoreactivity had the lowest fall rates. So again, we think that these alterations in blood flow to the brain have important implications in terms of gait and falls. Yeah?
Did you find the same association using the vasoreactivity of the blood flow as a continuous variable rather than to dividing quintiles?
We did. Yeah, thank you. I mean, that's a very important issue. We did. The overall model was significant, but we were more interested in dividing, into seeing whether it was a nonlinear.
Yes, in general, in clinical studies, we try to divide in quantiles to look for cutoff that may have clinical applicability in the future. Yeah.
Well, the other reason for doing quintiles is that it was nonlinear.
Yeah.
As you can see here, it's nonlinear. So that was more of a linear regression,
Sure.
And it didn't. I mean, it was marginally significant when you do a linear regression. When you begin to divide it out, you see that it really falls out in a nonlinear fashion, and for that reason, we present it this way. This is complicated, but we also looked at the relationship between cerebral vasoreactivity and gait speed. The overall model was significant, such that the higher the vasoreactivity, the faster the gait speed. And then we also did, so this was a similar type of analysis. We did an overall model, and then we broke it into quintiles. But leave it, I don't want to dwell on that. 2 of the quintiles here showed that there was a significant relationship between gait speed and vasoreactivity. These tend not to be linear, so I wanted to get away from that in my analysis. So what is finally, what is the evidence of association between cardiovascular risk factors and frontal subcortical symptoms? And just to complete the story, I'll show you some data there. Here we looked at the Framingham Index as a cardiovascular risk score, and looked at the percent of individuals with impairments in executive function as a function of the cardiovascular risk score. And you can see here that as the risk score increases, there is a nice linear increase in the percent of individuals who have executive dysfunction on a TRAILS B type of test. This is a test where you ask the subject to connect the dots from 1 to A to 2 to B to 3 to C. So they have to do what's called set shifting and do alternating identification of a number and a letter. So we saw a nice relationship there. And in another study, we again divided systolic blood pressure into quartiles and looked at the risk of impairment in trail-making B, and saw that the odds of being impaired in trail-making B increased markedly with elevations in blood pressure. So again, evidence that hypertension is a bad thing, not just because it causes big strokes and heart disease, but it also has profound effects on executive function, which is, we think, a frontal lobe function. We also looked at diabetics and again looked at executive function using 2 tests. These are clock drawing tests. One's called clock in the box, where you have to draw the clock in a box, and the second is a clock drawing test. And we divided those into people who scored high and low based on the median. And you can see that those who scored low on both of the clock drawing tests, either the clock in the box in white or the clock drawing test in black, had higher hemoglobin A1Cs, or greater risk of diabetes. And those who scored well in those two tests had lower hemoglobin A1Cs. So even within a group of, this is all within a group of diabetics. So again, diabetes seems to be associated with executive impairments. And we looked also at some data from the Charleston Heart Study, which are longitudinal data for the relationship between hypertension and motor function. In this particular study, there were 3 different measures of function: the Nagy index, the Roslow-Breslau scale, and the Katz ADL scale, which all basically look at lower extremity motor functions one way or another. And we divided the groups first by those with normal blood pressures or hypertension, and at baseline, their performance on these was similar on these 3, and the baseline was 1984 to 1985. We also divided the groups by those with normal blood pressures, controlled hypertension, and uncontrolled hypertension. With a Nagy, there's a tendency for the uncontrolled hypertension to have lower baseline scores in these. But at baseline, there were similarities among the 3 groups. We then followed them longitudinally through 3 waves of the study from 1984 to 1993, so 9 years. And you can see general declines in these scores over that period of time. But those with hypertension, shown in the red lines, had the most dramatic declines compared to those with normal blood pressures. And then finally, we divided the subjects into those with normal blood pressures, shown in the black lines, those with uncontrolled hypertension in the red lines, and those with controlled hypertension in the blue lines. And there was a trend toward a modifying effect of the controlled hypertension, such that those in the blue here were in the middle between the severe decline in those with hypertension that was uncontrolled and those with normal blood pressure. So if we treat the hypertension, we don't completely ameliorate the decline, but we modify it to some extent and reduce it. So these types of observations about the relationships between cardiovascular risk factors and these symptoms of slow gait, executive dysfunction, and even depressive symptoms led us to question whether the concordance of these 3 symptoms was a specific phenotype that described many of our older patients. Because those of you, most of you probably see patients clinically. Many of our older folks look like this. They have slow gaits, executive dysfunction, and depressive symptoms. So we asked whether these impairments aggregate together and whether they are associated with cardiovascular risk factors. And here in our database, we looked at the individuals who had 1 of these impairments, or 0 impairments, 1, 2, and 3 impairments, and correlated it with the level of systolic blood pressure. And sure enough, those who had more impairments had higher blood pressures, suggesting that, in fact, hypertension, you know, we traditionally think of hypertension as affecting large blood vessels and causing big dramatic things like strokes and heart attacks and MIs and vascular disease. But it looks like there's some subtle effects of hypertension as well on these 3 symptoms. And then if you've got all 3 symptoms together, you are more likely to be hypertensive. So we went further to say, does such a cluster of symptoms, or a phenotype, exist within our population of 800 community-dwelling elders, called the Mobilized Boston Study? As I've mentioned to you, we have assessments of their physical, psychological, vascular, and musculoskeletal functions. And we hypothesized that these 3 symptoms of slow gait, depressed mood, and executive function would be found together, or would cluster together. And to examine that, we used what's called a latent class analysis to see whether there was an aggregation of these domains. And sure enough, there was. I won't bore you with the details of this analysis, but the bottom line is that in 17% of our population, these three symptoms aggregated together. And in fact, in now other population studies where we're beginning to look at this, anywhere from 11 to 17% of the population have this combination of symptoms. So we do think that, yes, a phenotype of sort exists. And the question is whether this is related to some treatable conditions. So we asked, do those with this cluster of symptoms have increased cardiovascular risk factors? And the answer is a pretty definitive yes. When